trying to present myself.
Yeah, thank you. Thank you. Thank you for the opportunity to be here. So today we'll be talking about DNA diffusion. So, sorry, my name is Lucas Ferreira. I'm from Pinel Lab at the Harvard MGH. And today we're going to talk a little about how you adapt DNA diffusion or the diffusion to synthesize new regulatory regions. So at this moment, probably everybody has been heard about like this possibility to use this text to image models. And basically with this, you can have like a prompt, like an astronaut riding a horse, and you're gonna see that this image start procedure from a block that looks like a basic block until transforming the high resolution image, and you have like a very nice image of astronaut riding a horse. So at the Pinel Lab, you have like this artistic thing, but you don't wanna do any Mona Lisa, you want to try to paint DNA. It's not just DNA, you want to paint DNA that has self-specificity in it can represent regulatory elements. So how we try to adapt these diffusion models to work with this? So the way I show this, we are using image. And first thing is convert the image, the DNA image. And the way we did this was using a Halt encoder, but trying to have a Halt encoder that has normalization between -1 and 1, not a binary matrix. So, okay, now you have an image. And the next step is understand how the diffusion works. The diffusion works like in this 2 steps. The first one is destruction, destroy the sequence. And the way we destroy the sequence is adding noise from the image until we get like a totally messy and white noise image. So we do this step by step until you have this process destroying the image. In the second, we didn't learn anything about the creating image when you do this. But if you do the reverse process, it's basically you're trying to have a function that can learn once you have an image in a given step. We train this function to try to deconvolve and extract the noise from that image. So in this idea here, when you have your vector with this 200 base pair image, you try to extract exactly where is the noise. And if you do this from the noise until back again, you can recover the original image in the same way they do with the Midjourney, for example. So what do you use to train this? In this case, we use like you're trying to create like a regulatory elements that come from human chromatin accessible sites, and you use these three different cell types: the K562, GM12, 8780, HaptoG2, and we separate this data in validation, in test, and training. And the idea here was we need to approximate that function that can deconvolve from noise until back to the normal image. And then what you do, we get the each steps that we have, you're gonna have the image with a charger with noise that we add previously in the forward process. So now we have like some Y in this equation that we can approximate and try to, from the image, extract just the noise of that, since we added that noise previously. So we do this iteratively, but here you see that we are not learning anything about any specific cell. We need adding something on top of this, and what you do is adding these embeddings that basically get the sequence label and try to inject this in this structure that we call UNet. And the UNet is a very nice structure that can do image segmentation, but in this case we're doing noise segmentation. So it's a combination between convolution and attention models that can very nice do the job. So okay, now you have a model that can generate these 3 different labels that we call prompts here. And now we need to check if this model is working. The way we do this, we try to go with this in silico validation using different metrics. The first one is checking if you are memorizing this through a BLAT. And then we go check if the TF motifs, if the vocabulary that this sequence, if this model is learning is correct. And then we try to check accessibility. Look, Chrome BPNet again for issue here. And we have Informer. That's another way to predict accessibility and gene expression as well. So, okay, just to go very fast through this metric, the first thing is showing if you are memorizing or not. So this is just like a pairwise comparison that we blend the sequence against the training data set. You're showing that the similarity between the sequence that are being generated for the model are very low. It means like just 10% of sequence has at least 50% similarity. It's even lower than the train and the test data set. So it doesn't look like you're memorizing anything. The next step, okay, we're not memorizing, but what you're learning here is just like the network doing some trick to learn this. No, you're showing that actually you're learning some motifs that are interpretable. The way we do this, we generate sequence, for example, for the cell type C. Imagine we have this, and we do a motif scan on this. We do a motif scan in the train sequence as well. So now we extracted these distributions that are the motif distribution in the train set in the sequence that we generated. And now we can try to match the distribution of these motifs between these different cell types. And what you observe in general when you try to match this, and the way we match this is using KL divergence, we see that in general, when you condition the cell in a specific cell type, they're going to match with the motifs of the train set for that specific cell type. This is just a toy example, but if I show the real data here, it's going to show that this diagonal matrix is showing that always that I do this pairwise generated train with the same cell label, they're gonna have like the highest KL divergence, the lowest KL divergence, showing that they are similar. It's showing that we have very cell-specific motifs. So another way to show that motifs make sense is looking this by eye, and you check that during the diffusion steps, you have 50 steps of diffusion. In the step 30, we start to have like factors already well known in the field, and we are avoiding another factors that can not be cell-specific. So the next slide is okay. You have like now the motifs, and now you want to know if they are accessible. So you go for Chrome BPNet, and now you're asking, for example, if I generate using our diffusion DNA model sequence for GM cells, are they accessible or not by in the silico validation? So we show that if you compare the trained, the generated sequence, if you generate sequence for GM, the sequence, they are actually similar. If the models are predicting are the same. So again, we are doing accessibility, generate accessibility. It just looks like cell-specific in the same way that the trained data set, the test is. Okay, it's not just for GM. We find this correlation. Looks like all the labels are being learned for these 3 cells. And not just Chrome BPNet showing this. Informer, that's another state-of-the-art predictor for accessibility, is showing the same trend, showing that we are able to generate this accessibility. So here is a very important slide. It's basically showing that, okay, we have accessibility, we have motifs, but what's happening if you try to generate gene expression in the cell type that, for example, a gene was not expressed by adding these motifs that we, sequence that we generate in the DNA diffusion. So in this slide, I'm showing that, for example, in this cell, in the Y type, we have like enhancer for K562, you have expression for GATA1, but we don't have this for GM cells, we don't have expression, but when we introduce one of the sequence that we generate with DNA diffusion, now we have higher accessibility in this region, and now we have expression as well in that region. That's showing that informer is kind of indicating that that sequence has a potential of regulation. So in this place, we know that was a previously K562 enhancer, but if you're trying to find new GM sites that was not described before, we try to tiling this same sequence in the whole locus of GATA1. You find, like, again, we rediscover the same site, but same time we find another regions that when we put the sequence, we have like a boost in the expression of GATA1 that's represented for the red peaks, showing that the expression is going up. So looks like we are able to generate expression by in silico as well. So next slide, the conclusion is the next step. This is in silico validation. Now what you want to try is have like some people validating this experimentally. So you're gonna have like this testing the potential of the sequence generate expression in prior arrays using GFP, and then you're gonna have the accessibility in vitro using MIA as well, and try to have endogenous substitution by getting some locus with collaborators that has some disease correlation, and try to add sequence to regulate that locus. And we want to expand this model for more cell types as well. So the conclusion, DNA diffusion can generate sequence in the cell-specific manner. It has a potential to regulate gene expression. So thank you to Pinel Lab, all the collaborator. Think Open BioML, that's a very important, is like 50 persons that are helping us like in the open source to develop this, giving resource in new researchers. And thanks Walter, Emily, and Kundaji for the Chrome BPNet software. Thank you, and visit my poster to know more details about this. Thank you.
Thank you. Okay. We have time only for one question for these short talks.
Thank you. That was a really nice talk. Very interesting work. I have a couple of questions about the method. The diffusion process standardly pushes the data to an isotropic Gaussian noise. Is it safe to assume that DNA sequences are distributed in the same manner, or would you consider a discrete diffusion type of process in future work?
Yeah, I think the way we approach this is try to discretize the sequence to have the distribution that goes through this. So when you see the first image, you see that I'm not using a binary hot encoder. I'm using a hot encoder that has a boundary -1 and 1. So when you have 0, you have the random probability to each nucleotide. So we're basically random sequence. So that's what looks like working.
All right, thank you.